Okay, hi, Roel. Thank you for joining us. We're here to discuss your exciting Gairdner Award. And I just want to start by saying that, for many in the audience, WINTs are something that we read about in textbooks, and we take for granted how they work at a great level of detail. But this award was given for your seminal discoveries of WINTs when they were completely unknown. And one thing that fascinates me about these discoveries is to try to understand the genesis of them. And this is a particularly exciting story where you discovered WINTs when you were trying to understand the formation of breast cancer from a mouse virus. And I wonder if you could bring us back to those times and explain how you discovered the first WINT in a mammal, and give us a flavor of how exciting those times were.
Yes, thank you, Steve. Yes, it all goes back to an early interest that I had in how breast cancer would arise in a mouse model. And there's a retrovirus in the mouse called the mouse memory tumor virus. I had been working on that virus even as a PhD student in Amsterdam. But then I did postdoctoral research with Harold Farmers at UCSF, starting in 1980. And at the time, we were interested in pursuing this model that the mouse memory tumor virus would act as a mutagen. It would insert itself as a DNA copy into the genome of cells, host cells. And by that insertion, it would possibly activate a gene that would be in the vicinity. And what we did was to clone molecularly the inserted copy of the mouse memory tumor virus DNA plus the adjacent DNA. And then in the adjacent DNA, the hypothesis would predict that there would be a gene of interest. And that is how we found, indeed, the first member of the WINT family, called WINT1. And then subsequently it became clear from a lot of work done by many different labs that the gene was widely implicated in development, embryogenesis. It was also highly conserved in evolution, so you could look at WINT sickling in almost any animal species that you would have an interest in. And then at the end, coming back to cancer, it turns out that the WINT pathway, as we now call it, was widely implicated in cancer, not only in breast cancer, but also in major forms of human cancer, including colon cancer and many other forms of human cancer. So it was indeed a story that started with cancer. It then branched out into development and evolution and all kinds of other interesting biological areas, but subsequently it did all come back to cancer, and then more recently also in the control over stem cells.
So it's really a remarkable series of discoveries, and I just wonder, back in those times in the early 80s, when you first described what you call INT1, there was no homology to known proteins at that time, and this must have been a point of frustration for you to not know what it was that you were studying at that point. Can you tell us how you thought about it at that time?
Yeah, that's interesting. It's a sort of a mixed bag. When you identify a gene and you determine the sequence, you always hope for some kind of homology, right? Because then you can indeed deduce from the homology how it would actually work. But we didn't find any homology. And with hindsight, I now would say, yeah, we were very lucky that we were finding something new and exciting. We did know from the sequence that it was a protein that was going to be secreted from cells and likely acting as a growth factor. And that turned out to be the case. It is now clear that WINT signals are among the major signals that operate in tissues and development, etc. So yeah, it took a while to get insight into how it would actually work. And a lot of the understanding of wind cycling didn't come from any biochemistry initially. It all came from genetics, and in particular genetics in fruit flies, where this whole pathway operates as well. And from the genetics in the fly, people, including our own lab, have been able to elucidate the pathway, the core of the wind pathway, as it is now called.
And when you made that pivot to begin working on Drosophila, you mentioned that you had done your graduate work and then your postdoc and your early independent lab work on tumor viruses and trying to understand breast tumors. But then you made a pivot to work on Drosophila, which was completely, an amazing decision on your part in retrospect. And how did you decide to do that?
Well, it started with an experiment that a graduate student in my lab did, and that is to look for copies of the Wind gene in the fly genome, initially by simple blotting and looking for sequence homology in the fly. And he found at the time that it was actually present in the fly genome, but now as a gene that was implicated in fly development, a gene called wingless. And then, we realized that since there was so much known about fly development and there were these genetic approaches, that we could maybe understand what the wind pathway would be, but now studied in the fly and by using mutants. And that was an initial realization, but not much. I was not able to do a whole lot until I started to acquire more and more knowledge about fly genetics. And then I moved to Stanford, where there was a lot of fly development being done in the Department of Developmental Biology. And that made it possible for us to really become a full-time Drosophila lab, which we did until maybe now 10 years ago. And then I switched actually back to studying mammals and cancer and stem cells.
Sure. And this was, I'm guessing, the first link between a developmental pathway and cancer, right? We take that for granted now, that cancer is a process in part caused by developmental pathways gone awry. But this was, I imagine, the first link to development, between development and cancer.
I believe so, yes. I have to say, at the time, there was a lot of thinking that the oncogenes that you are also very familiar with, that were all discovered in mammals, also using other retroviruses, they had homologs in the fly. And there were also other signals that members of the TGF-beta family, copies of those were also found in the fly genome in a gene called DPP. Mm-hmm. So it was a realization that came about around the same time that we found the homology between wind and wingless. And that was indeed a very exciting period because the tyrosine kinase is also widely implicated in cancer. They had homologs in the fly, so people could see that by combining those two fields together, cancer and fly genetics, you could learn a lot about how these things would actually work.
Very exciting. And as you indicated, now the Wnt pathway is so integrated into our understanding of cancer and so widely mutated in many different cancer types. And there's been tremendous interest at drugging this pathway as a potential cancer therapeutic strategy. So what are your thoughts on that? How far away do you think we are? And what do you think is needed for us to capture that potential?
Yeah, that has taken a long time. People have been very hopeful that by interfering with wind cycling in cancer, you could actually do something for therapy. There are some reasons that it was slow, but lately there are some hopeful signs. There are now small molecule inhibitors of a component of wind cycling. The gene is called porcupine. that are actually very effective in blocking the wind signal itself. Not so much the interaction of the wind components in the nucleus. But those molecules, they may actually have effect in a context where the environment of a cancer cell is going to be important. And it now is becoming clear from a lot of work that in this whole process of where cancer cells are proliferating and the environment, and in particular metastasis of cancer cells, that if there is a wind around, that these incipient tumors and metastasis would actually grow better. And that would open up the possibility to block wind signaling at that level and maybe interfere with the growth of tumor cells in particular locations, including during metastasis. And so there's reason to be somewhat optimistic, or more optimistic now than maybe 10 years ago. And there could be all kinds of other things down the road where we could actually take advantage of this extensive knowledge of the signaling pathway to block the growth of tumor cells, including human cancer.
Yeah, I think these are very exciting developments, and we look forward to continued progress in this area. And finally, can you tell us, you've won many awards now for these discoveries, and I wonder if you could tell us, what does the Gairdner Award mean to you?
Yeah, so the value of an award, a prize, is always coming from who else has been getting the awards, right? So if you go down the list of previous Gairdner Award winners, that is a very impressive list of people. And and that is why this award is, to me, it meant a whole lot. I have to admit that when I got a phone call from Janet Roeland, she's the president of the Gairdner Foundation, I was very excited because I knew that it was a very prestigious award. And also the other people who got the award this year have a strong track record. So yes, it is something that is exciting. And hopefully there will be a ceremony at the end of the year, but it already has been postponed to next year. But yeah,
I see. Well, I hope that will happen in person. And I just want to say again, Roel, congratulations on winning the Gairdner Award, and thank you for spending time with us today.
Okay, thank you, Steve. Thanks.